during this tragedy, and it's just, I know you guys are just great lights for this city, and I'm glad that you're doing what you're doing. Okay, let's just be clear. I am not a preacher. I'm more of a biology teacher, so this is going to seem like a biology class this morning. Is that okay with everybody? I hope in the end we're going to wind up giving God glory for what He has done in His creation. And actually, the last 3 years have just been amazing because our science technologies have increased greatly. And the more they increase, the more it shows how God had to have created. There had to be some kind of intelligence behind all of this. And I'm going to try to show you a little bit of that this morning. Is that okay? Say yes. Okay, good. I've still got you here. We're going to be looking at the works of the Lord. I'm going to look at 3 different areas this morning. First, we're going to look at the language that's being used in the science literature. Then we're going to go to a little bit of engineering in organisms. Engineering means there was an intelligence there. And then recent human-chimp research is just one of the greatest things we saw this year come up in the research. So that's what I'm going to cover this morning. We'll see how we can do. The language in science is changing. They're starting to use words like complex, machines, engineered. We're seeing all these kind of words. This book just came out recently in 22 by a medical doctor and an engineer. They worked together to show how the body had to be designed by an intelligence. And it's just really, really cool. One of the things they look at in that book is the circulatory system, and they show how there's so many problems that could go wrong in the circulatory system. Its job is to carry oxygen to every cell of your body. You've got 30 trillion cells in your body. And so that means you're going to need that much oxygen every second. 100 million trillion molecules of oxygen every second. Your body has to have that. But the oxygen's on the outside of your body. How do they get that into your body? Well, in your lungs, you have these air sacs, alveoli. Now, to get that much oxygen in your body, you have to have a surface area that's equal to a half of a tennis court. That's a huge amount of surface area. But with 300 million alveoli in each lung, those little air sacs, that's enough surface area to get that much oxygen every second into your body. Is that not incredible? Just incredible design there. And then the blood system surrounds those alveoli, and the blood is carrying hemoglobin made of iron. But we have to get iron from our food, but when we eat the food, the iron is toxic to our body. So your digestive system has to work in coordination with the circulatory system. It can't take in too much iron or it'll be too toxic. But once it takes it in, it makes a little compound called transferrin to carry the iron in the body to the bone marrow so that it doesn't be toxic to the body. So there's just so many different problems that could be encountered along the way. It just shows there was a lot of engineering going on in there. So it shows there had to be an intelligence. It's just amazing. If you want to read about this or look at this, they have videos now called Secrets of the Human Body online, where these 2 guys, these engineers and medical doctors, talk about the body and how it is engineered. This is their first episode. Their second episode is out already. It's called Living in a 3D World. You ought to go there and look at there. Just look at secretsofthehumanbody.com. It's just really fun to look at all of that. This was an article put out by Harvard University. You can see that back in 21. And they were talking about a bacterial cell. This is just a bacterial cell. This is its flagella, and it propels itself through the water by that flagella. And to do that, it uses a motor. Notice they call it an engine right here. It's a motor that turns around. And they call this thing here a clutch. When that clutch is engaged, it turns the flagella and propels the bacteria through the water. And with just a less than a millisecond, it can unclutch it. It can disengage that clutch to where it's still and will not be moving. But what's really interesting is the language that they use. Listen to the language. The key mystery of the flagellar motor lies in the propeller-like movement. Much like man-made motors, the molecular details illustrate an interesting finding. The machines that people invent often work off the same principles as the machines that drive small biological life. Isn't that interesting? They're saying it took an intelligence to put this together. It's just really fun being able to see that. Now then, I'd like to look at a little bit of engineering, and this was brought to my attention just this year. In fact, these studies were done in 2025 to show all of this. This is in Asian rice. You grow rice in warm temperatures. Has to be warm temperatures. They decided to start seeing if there was any rice that would grow in cooler temperatures. So they took 31 varieties, started growing them in colder temperatures. In fact, they took them from this part of China and took them way up here to grow them in cold temperatures to see if they would produce rice, produce seed. And so they found one variety called KD8 that after 4 years of planting it up in the cold temperature, it finally started producing rice seed. So in the evolution world, they would just say this evolved. This particular rice plant gained a new trait, it gained new DNA, and it was able to now make seed. And that's been the thinking all along in the people in the biological world. The theory of evolution has been dominant in causing us just to think everything just evolved or just changed. Well, these researchers decided to go a little bit further and look at the DNA and find out exactly what did happen in these rice in this rice plant. It's just this one plant that was doing that. And what they found was there was one gene called the ACT1 gene on the DNA, and that was the trigger to help get this rice to start producing in cold temperature. Now, normally there are tags put on that gene to stop it from functioning. And that gene didn't function during warm temperatures, during the normal life of the rice plant. But in cold temperatures, those tags were not put on there, and that activated the ACT1 gene. So here's what happened. When the cold temperatures got cold, there was this gene, MET1B. Its job is to put tags on AGT1. But when the cold temperatures hit, it was reduced in its ability to put the tags on, so no tags were put on, and that caused AGT to start functioning, and when it started functioning, it caused a whole cascade of events that actually led to rice being produced. So there was no evolution going on here. The mechanism for this rice to produce seed in cold temperatures was built into the DNA already. And just when it got in cold temperatures, it just caused this one gene to be downregulated, which caused another one to be upregulated and help it make seed. It was a built-in system from the beginning. It wasn't anything new that the environment caused. It just shows us that God built things into every organism to be able to meet or adapt to different environments. Isn't that cool? Yeah, listen to this. The author said the biggest challenge was convincing reviewers that there were no genetic changes driving this adaptation, which is why they had to consider extensive genetic tests to rule that possibility out. It doesn't occur by evolution. It's occurring because God built that into every organism. Isn't that cool? Just really cool thing. So this is what we call epigenetics. The DNA stays the same, but it just is modified just a little bit. So here we had an external stressor. It caused an internal mechanism to occur that modified the DNA. It removed these tags, and it was able to operate. So that's epigenetics. The environment can cause an organism to to switch something on or off. That even happens in our bodies. In our bodies, if we undergo stress, our DNA can switch things and turns things on. We can even have genes jump from one chromosome to another to turn on a particular gene. God built us with that ability to do that. So these are built-in mechanisms in the DNA for adaptation to different environments. That is engineering. That is an engineering thing from God. Here's another example of bacteria. Bacteria usually feeds on glucose or ribose, common sugars they find in the environment. And in fact, these bacteria can even count every glucose molecule that it passes. And if it sees more molecules on this side of it, it can turn and go towards where there's more sugars. So it's a really unique system that they have. Well, these researchers decided to remove all sugars and starve the bacteria and giving it only beta-glucosidase in the medium. And so they put these bacteria in this media with beta-glucosidase rather than glucose, and they were starving. They were not growing at all. But after 7 days, one bacteria started to grow and divide and produce a whole population. At this point, evolution would say it evolved. It gained a new gene to where it can now survive in this particular environment. And so this is a lot of research done on this. You can see all the papers that were made because of this. But what actually happened was this. The bacteria already had the gene that would metabolize beta-glucosidase. So that's the abbreviation for beta-glucosidase. Does that look like bagel to y'all? Yeah, so let's just call it bagel. It's a lot easier to say if y'all do that. But here's the DNA, parts of the DNA, and as they got to looking at the DNA and what was there, there's a gene right here that metabolizes beta-glucosidase. It could live and feed on that particular sugar. There was also another gene right here, right next to it, that would bring beta-glucosidase into the cell. And there was another gene right here that would detect if there's beta-glucosidase in the environment. It's a particular protein that would embed in the membrane of that bacteria and detect if there's any beta-glucosidase in the environment. This is called an operon when you have several genes all together. They're all made at the same time, and they have a promoter over here. Well, without the beta-glucosidase and in normal conditions of glucose, the promoter is shut down from the rest of the genes, and it doesn't work. Those genes aren't turned on. But when beta-glucosidase is in the environment, and if let me first talk about this protein. This is a certain protein that binds right here onto the DNA and causes this loop and shuts this thing down so that the promoter can't make those particular proteins. So that thing's always turned off. Well, this bacteria that was starved for a long time in the presence of beta-glucosidase, what happened was whenever a bacteria is starved, it starts making lots of cyclic AMP. So it just makes lots and lots of cyclic AMP. And the cyclic AMP caused a couple of things to happen. It caused the cyclic AMP bound to the promoter here and caused this thing to open up. When it opened up, starving also caused the production of this particular protein, which made a transposable element. A transposable element is a gene that's going to jump. And it jumps from one chromosome and will jump into right here, into where that SID was at. So now the promoter is connected to all of these, and it can make all these proteins and live on beta-glucosidase. So it's a built-in mechanism. Do y'all see that? God built in every organism these mechanisms to adapt to whatever environment it is in. It's just remarkable. The woolly mammoth, they finally sequenced the DNA of woolly mammoth. And sure enough, It's an elephant. It's kin to the African elephant, the forest elephant, the Asian elephant, more kin to the Asian elephant. But the difference in the woolly mammoth from the elephants, it's got 87 genes that were deleted. You can see all these elephants have this particular gene, but it was deleted in the woolly mammoth. In fact, 87 were deleted. They call them deletions or indels. An indel is an insertion deletion. It inserts something into the gene that breaks it so that it doesn't work. And so the woolly mammoth came about just because it had a bunch of broken genes, but it was able to live in a certain environment. Anytime you put an organism in stress, it's going to move genes around in order to be able to adapt to that stress. And that's what happened with the woolly mammoth. But it's still an elephant. It's still in the elephant family. Y'all catch that? Yeah, kind of fun. So it seems like all organisms have this incredible amount of built-in genetic variation that helps to adapt to all kinds of stress and situations. The DNA of organisms doesn't change that much. There's a group of organisms, they just have a lot of variation, in a particular family. So this is showing the engineering of our Creator, having things in place already for different things to occur. Y'all like all these vegetables? Broccoli, cauliflower. I'm not a fan of all of these. They're all from the same plant, from the wild mustard. Man has taken the wild mustard and bred it and did breeding practices to select for certain traits, and we're able to select for all of these. And so it's all the same plant, but you can see the amount of diversity that is in the DNA. That's how God made every organism, with a lot of diversity so that it can adapt to different situations. These are the silver swords in Hawaii. If you've ever been to Hawaii and seen these plants, this plant, this plant, this plant, this plant, this plant, this plant, they're all the same plant. Some plants are just herbaceous in one environment, but in another environment, a gene will turn on that'll make woody tissue in that plant. But all of these are the same plant. I even have a hybridization matrix here which shows how they can interact with each other, or they can breed with each other and produce viable offspring. So you and I would think, no, there's no way this plant could breed with this plant and produce viable offspring. But they do. It's just God built in a lot of diversity. Are y'all getting my point here? Yeah, it's just a lot of diversity. All of the Drosophila, they're all the same species. They can interbreed with each other, but they've got different characteristics. We have a lot of diversity built in to all organisms. These are the cichlid fish in Africa, in the lakes. These are the ones in Lake Tanganyika. These are the ones in Lake Malawi. They all came from a common ancestor a long time ago, but in these different lakes, they adapted to the different parts of the lake and took on different forms. But you can see in the other lake, they almost have the same forms that they had in that lake. They had the same original genetic material from their ancestors. And they diverged in different lakes, but they diverged the same. So that just shows there's a lot of built-in diversity within a species. Can y'all see that? Yeah. Now, evolution, when it does its thing, it will give them different names, and y'all can see all the different names that they give to these scientific names. But in actuality, they can still interbreed with each other and still produce viable offspring. It's just kind of really, really cool. There's Darwin's finches. They say it's 14 different species. Now they've reduced it down to 5 different species. They can all interbreed with each other. And they just have different characteristics for different parts of the Galapagos Islands. So it's just kind of fun. Even in the wolves. In the wolf, if our wolf is the ancestor to all the dogs, look at all the variation that we can get just from the wolf population. A lot of diversity that God built in to all of those. They actually took a wolf and crossed it with the poodle. Y'all want to know what they got? Yeah, they got these. They call them poodle children. Yeah, they all look the same. That's because The wolf has this genetic material, the poodle has this genetic material, and when they crossed them, they have both for every gene, one from the wolf and one from the poodle. Well, then they crossed these with each other, and look what they got. It's quite a surprise, isn't it? Yeah, you don't see any of those traits in those. They're hidden because the way genes work. We have different forms of alleles, and so that's what they've got. So in biology, we're relating kind. When God said, take every kind on the ark, he's really talking in biology terms, the family level. The family level. Yeah, family, genus, species. And so the family level is the same species. You have the cat family, the dog family. We never see any organism go out of the family level, which means that's one species of that family. So we've got the whale family. We have dolphins and whales in the same family. They can interbreed and produce viable offspring. So there's a lot of diversity that God built into each kind, is what we're finding. So I hope you're seeing information architecture in organisms. All we have to have is a bunch of genes with at least 2 forms of each gene, and you can get so much variation that can adapt to different environments. In every environmental situation, different conditions of genes allows for a phenotype that best fits that environment. In addition, there's other built-in mechanisms, epigenetic mechanisms, like jumping genes that also cause different traits. Which brings me to Genesis 2. Not Genesis 1, but Genesis 2. Listen to what he says. Thus the heavens and the earth were completed in all their host. By the 7th day, God completed his work, which he had done, and he rested on the 7th from the work he had done. Then God blessed the 7th day and sanctified it, because in it he rested from all of his works, which God had created and made. We can trust that God was the creator of all of this. Even evolution today, they've not said a word since 2012. Since all of our new genetic technologies from sequencing genomes have come about, evolution doesn't know what to do with it, because we're able to look at genes, we're able to test their theories, and it's just showing a lot of diversity in each species, and in different environments you get different looking organisms, and that's all it's showing. So it's been really fun to be in this world, just really, really like fun. Last thing, are y'all holding up okay? Okay. Last thing, I had to talk about it. This only came out a couple of months ago, but it's really big. Y'all know in the evolution world, we're considered to be our closest relatives are the chimpanzee, and we're 98.5% the same. Our DNA is 98.5% the same. Well, these guys, they're not creationists. These are evolutionists. They did a complete study. It was a really, really good study. I'm not moving here. Okay, y'all may have to help me move slides. I can't move it. Oh, there we go. Went too far. In previous studies where they've compared human and chimp DNA, they did incomplete sequencing. When they tried to see what the chimp DNA was, they used the human DNA as a scaffold, and they'd take a piece of the DNA and, that matches here, and another piece and that matches here. And they only used the human part as a scaffold, and that made it look like a human. That made the chimpanzee like a human. And we've always get 98% similarity. Well, this study, they did the study according to very high standards. They had proper filtering protocols, hand curating sections that did not behave. They worked with them, and they didn't use the human DNA as a scaffold for all the others. And they looked at all of the great apes to see how close we were to the chimpanzee and others. There's their data. Now let me show you how it looks. The orange here, when you're looking at the orange, that's within a species. So within the human population, we're about 3% different And I can look around this room and yep, we're quite a bit different. And so we're 3% different. And so the chimpanzee, among all chimpanzees, they're about 8% different. Same with the bonobo. The gorilla, there's about 14% difference within the gorillas. So that's what the orange means. Now then, the dark blue is where they compare humans to apes. That's the dark blues. And the light blue is ape to human. So they did it both ways to make sure they could compare them correctly. And look what they found. Between human and chimp, we're 15% different. We're really different. We're 15% different. We're 15% different from the bonobo. We are 20 to 30% different from the gorilla. I know we've met a couple of guys that we think that probably, no, we won't go there, yeah. The orangutan, we're 20% different from the orangutan. So what does this mean to all of us? Well, we're only 84, 85% identical. Not even close to the 98 to 99% identical. And this data is not highly advertised, nor is the data readily noticeable in the paper. The paper was 300 pages long, and you really had to dig to find out what they were saying in the paper, but it pretty much comes up, we're 15% different. We should expect a high similarity. The chimpanzee and us, we have the same body plans. We can both kind of walk upright. They walk a little different than we do, but we can walk upright. We have 4 limbs and everything's pretty close. So you'd expect some high similarity there. So our creator would have used many of the same mechanisms. So for now, we're 15% different. And that's really, really good to know. We're no longer 1% different from a chimpanzee. Now, I know you're going to think 85% similarity seems to indicate that we had a common ancestor. 85% may seem kind of close to you guys, but then you have to ask the question, how close are we to other organisms? We're about 80% the same as other organisms, particularly a cat. In biology, we dissect a cat in lab, and if you look at the inside of a cat, it looks just like the inside of a human. Just almost exactly alike. So we're 85% the same as a chimpanzee. We're 85% the same as a cat. We're 85% the same as a pig, as a horse. The horse insides looks just like our insides. What God does is He's used the same DNA in all organisms, the same DNA, because we all have to breathe, we all have to eat, we use the same mechanisms. But then there's that 15% that's just human. There's that 15% that's just chimpanzee. There's that 15% that's just cat. Do you see what I'm saying? Yeah. So don't let the 85% throw you that we're really close. It just means all organisms are like that. They compared the DNA of a mouse, a zebrafish, and chickens. They compared their DNA, and they're looking at gene families. And so the bigger the circle, the more gene families that they're looking at. Humans, we have about 63,000 genes. It keeps increasing every year as we find out more about our DNA. But so that gives you an idea. If we have 63,000 genes, you can see what we're looking at here. And so when they compared these 3, the mouse, the zebrafish, and the chicken, what you see is green here, that's the only part of its DNA that's just chicken. It's just chicken. I don't know what color that is, whatever color that is just found in the mouse, but not in the zebrafish or the chicken. And so you can see the mouse has a whole lot of DNA that the others don't have. It's just really unique to a mouse. But then when you put them together, this group down here, that's how much DNA is shared between the mouse, the zebrafish, and the chicken. That's a huge amount of DNA. So all organisms, we share at least 60, 70% of our DNA with the fish. We just share that much together. Now, we'll be closer to all the mammals than we will be to the bird or to a fish. But this is just showing you that we all share the same DNA. We all just have a thing. So to say we're 15% different from a chimpanzee is a big deal. Do y'all see that? Yeah, it's really a big deal. So, this is my last slide. God tells us this, that He did all of this. And He was, now that we can see the DNA, the complexity of it, it just makes me want to be in awe of our God. Listen to Colossians. For by Him all things were created in heaven and on earth, visible and invisible, whether thrones or dominions or rulers or authorities. All things were created through Him and for Him. Yeah. I'm done.